All right, and welcome to the second part of our B cell development lecture on Chapter 9. This will finish up the chapter. So B cells, as they finish their bone marrow portion of their maturation, when they get exported from the bone marrow, they're still not functionally mature. So they're still immature. They have to go through a final maturation process in the spleen. So if we look at, here we have from our bone marrow, let me get my pen here. We have our pro and pre B cells that will mature into an immature B cell, differentiate into immature B cell. That will then go and enter into the spleen, where if we zoom in here, we can actually see there's going to be two subsets of B cells in the spleen called transitional 1 and transitional 2 B cells. Unfortunately, they shorten that by calling them T1 and T2 cells. These are not T cells. This is a very unfortunate naming decision to call them that. These are T1 B cells and T2 B cells. So in the spleen, they'll go through it. They'll start initially as T1 and then differentiate into T2. And the difference between them is really all about different gene expression, as you might expect. There's also some different surface receptor expressions, as you can see from the image down here in Figure 9.7. And so they'll go through the difference in gene expression from T2. They will eventually go into the follicle and mature into the follicular B cells, or they may also be able to mature into marginal zone B cells, which we'll talk about a little bit later. The follicular B cells are the more common B cell that we've kind of mentioned already in this class. So closer look at the T1 and T2 B cells in the spleen. So the T1 subset is still actually undergoing some screening, and there is the possibility of a negative selection event. So if a T1 B cell is able to bind to a self-antigen with its receptor with a strong affinity, it will be induced to undergo apoptosis in response to this. So this actually just shows that there's this would be an example of peripheral tolerance, peripheral screening, because it's not happening in the central lymphoid, it's not happening in the bone marrow, it's happening outside in the periphery in the spleen. And it's believed actually that 55 to 75% of T1 B cells here actually will be eliminated by this process. So a substantial amount of negative selection of B cell development actually does happen in the spleen in this T1 subset. As it does transition to the T2 stage, it loses this ability. So if we look at this, you'll see from T1 to T2, we're going to lose the ability for this process. We're going to move from the perarterial lymphoid sheath, or the PALS area, into the follicle, where it will no longer be able to undergo negative selection. So once in this T2 stage, it is no longer capable of being induced to undergo apoptosis by binding to its antigen strongly. This is kind of like in the T cells when they become single positive development, they lose the ability to go through negative selection. Same idea here. What we do see in our T2 cells here is that they have to receive a survival signal through signaling of the BAFF receptor, this BAFF receptor. That's a very important receptor for all B cells, very important survival signaling. When a receptor binds to the BAFF receptor, when they receive that signaling in the follicle of the lymph node, those that do receive it will be induced to survive. So they'll be positively selected for mature B cells. Those that do not receive the signaling, as you can see on the left here, will not survive, will undergo death. Throughout an entire B cell development, the B cell life, they have to have this BAFF receptor signaling, and in response to that, that's going to turn on expression of anti-apoptotic genes that will prevent the B cell from undergoing apoptosis. So basically, it tells the cell to stay alive. This will be important for B cells when they get activated, which we'll talk about in the next chapter. This table just shows the difference in terms of the responses to strong B cell receptor signaling between T1 and T2, and then mature B cells, which are also known as B2 B cells. Yes, there is a B1 B cell. We'll talk about that a little bit later. And you'll see in this that the only subset that's able to induce apoptosis is our T1. And the reason why they're able to induce apoptosis is that they are not inducing expression of this BCL. So it's BCL-XL, that's an anti-apoptotic gene. Both the T2 and our mature B cells are able to induce that expression of that, and so therefore they do not undergo apoptosis. because they're expressing a gene whose sole purpose is to stop the process of apoptosis. The other aspects that we see between our T2 and our mature B2 B cells that we don't see with our T1 are things that are related to cell signaling. So we have increase in the diacylglycerol, that's the DAG, if you'll think back to when we talked about cell signaling, increase in calcium ion concentration, formation of lipid raft. These are all things that are going to help with cell signaling, allowing the T2 and the mature B cells to receive the signaling through that BAF receptor to induce expression of these genes that will prevent the cell from undergoing apoptosis. So after going through the T2 stage and maturing into mature primary B2 B cell, this is kind of the classical B cell that we think about with B cells, and they're going to migrate to the follicles of the lymphoid follicles, and they're going to start expressing high levels of both IgM and IgD. We've already talked about IgM a decent amount. IgD, remember, that is the one of the antibody classes that we don't fully know the function of. We just know that it's important in B cell development. So B2 B cells are expressing both those, IgM and IgG, on their surface. And at this point, they're able to recirculate between the blood and the lymphoid organs. So they're basically going to take on a functional mature B cell that's going to go circling around trying to find this antigen at some point. They'll be there to help respond to antigens, and they need T cell help to do this. We'll go over that in more detail in the next chapter. We kind of touched on it periodically throughout the course, but B cells, in order to get fully activated, will need T cell help. And they'll help in the process of immune response by producing antibodies, as we know. So their half-life in the periphery is about 4.5 months. So here's just a look at how the surface marker expression differs, again, between our T1, T2, and mature B2 B cells. So the membrane-bound IgM shown here is high D1, T2, and maybe a little bit less high, but it's still present in mature B2 cells. The membrane IgD is kind of starting very absent or low T1, intermediate in T2, and high in our mature B2 cells. So again, the IgD, we don't fully know the function, but it is important as the B cells develop into mature B2 cells. Again, we see that BAF receptor is higher in both our T2 and our mature B2 cells. Both the T2 and mature B2 cells are also going to be expressing CD21, which is the important co-receptor of B cells. That'll be important for their activation. It's also, if you'll think back, an important receptor for binding a complement portion. So complement that can help also initiate and allow for B cell activation to happen a little more readily. CD24 and CD93, you'll see are markers that are lost as we transition eventually to mature B2 cells, and they're markers that are more commonly found on immature cells. All right, so the follicular B2 B cells, those are the B cells we've kind of referred to throughout the semester so far, but there are two other subsets that also exist, and they're going to be somewhat different from our B2 cells. So there's the B1 B cells, and then there's also the marginal zone B cells, and you can see some characteristics of them in this figure here. So if we just look at the B1 B cells, you'll notice some things that are missing compared to B2 B cells. We don't have the CD19, CD21, so it doesn't have the co-receptor. Marginal zone B cells do have that. B1 B cells do not. They have the CD5 that is present only in a certain subset of their cells, and other things that are very different for them, if we look down at the table here, is that the B1 B cells are present in different sites on the body, whereas the B2 B cells are going to majorly be in the secondary lymphoid organs. Our B1 B cells are in different other areas, so peritoneal and pleural cavities around the body instead. They are able to respond to carbohydrate antigen stimulation, whereas our follicular B2 B cells might be able to, but not always. And our B2 B cells are going to have high levels of IgG, whereas our B1 is primarily IgM, maybe some IgG. They also do not require T cell help, which makes sense why they don't have the co-receptors there. Our marginal B cells, if you notice, are going to be more similar in terms of surface receptors to our B2 B cells, but they have the CD1 receptor here, which is going to be a little different than what our B2 B cells have, CD23. We'll talk about that in a minute. All right, so a couple of interesting things about the B1 B cells specifically. So they actually come from a totally separate developmental lineage than the B2 B cells, and we know that because if you have mice that have mutations that prevent them from forming hematopoietic stem cells, they will therefore not have any B2 cells that are going to come from that, but they still will have progenitor cells that can become B1. So B1 progenitor cells are still detectable in the fetal liver of those mice with that mutation. So they know that B1 B cells come from a totally separate lineage. They, like I mentioned, are going to be in different areas inside the body. They are constantly generated outside the bone marrow. B2 depletion, if we do that, we'll still leave functional B1 cells there. So they have a totally different developmental process than the B2 B cells do. The B cells that are present in the pleural and peritoneal cavities of mice, 30 to 50% of those are going to be B1 B cells. They are much more limited in their receptor repertoire, so in terms of what their receptor can bind to and the amount of diversity that comes in the receptor, much more limited with them. They will bind to microbial carbohydrate antigens. That's more commonly what they're going to bind to, with pretty much low affinity. And all of this should sound more familiar to our innate immunity, which is kind of what they're familiar to. So they're much more similar to our pattern recognition receptors present in innate immunity. One thing that's interesting is that during the developmental process of our B1 B cells, they will actually undergo apoptosis unless they interact with self-antigens, which is kind of the opposite of everything else we've talked about so far in terms of T cells and our B2 B cells. So they actually go through positive selection when they interact with self-antigens in the developmental process. So they really are kind of a different totally type of cell, even though they are still considered a B cell. So the third subset of B cell development is the marginal zone B cells. So some B cells will develop into these. These are named because they're found in the marginal zone of the spleen, specifically the white pulp outer region of the spleen. And they seem to be important and very specialized for responding to blood-borne antigens. And that's why they're going to exist in the spleen, which, remember, the spleen is going to be the first response for any blood-borne infection. These marginal zone B cells are going to help with the B cell portion of that response. So they are able to recognize both protein as well as carbohydrate antigens, much like the B1 cells are able to with the carbohydrate antigens. Some of them may be able to recognize this and produce antibodies without actual T cell help. Others will require T cell help. One thing that characterizes them from the B2 B cells is they're going to have lower levels of IgG and lower levels of the Fc receptor comparatively. What they believe happens in the process is that these marginal zone cells seem to be derived from T2 cells that do have a strong self-antigen signal through the B cell receptor, but also have binding of Notch ligand at the same time. So a combination of that is what will trigger the T2 transitional B cells to differentiate into a marginal zone B cell instead of a B2 B cell. So let's compare the B cell and T cell development process. There's a lot of characteristics that are shared between them. We didn't go over this, but in terms of the gene segments, both are going to undergo a rearrangement of gene segments to rearrange what that T cell receptor and B cell receptor is composed of and what it's able to bind to. That was the chapter that we skipped, but we just said know that this happens. There's also going to be a screening process they both go through to avoid binding to self. So both of them are going to go through a negative selection process where we eliminate any that have a strong self-reactivity. They also are able to produce small subsets that have a much more discrete function. So our B cells, we have our marginal zone B cells and our B1 cells, and the T cell, we also had small subsets from the T cell development that had different functions as well. And then there's also the production of the larger general purpose subsets, so our B2 B cells, G helper cells, our cytotoxic T cells, larger purpose ones that do the bulk of the work in terms of the major function of the immune system that we spent a lot of time talking about this semester. But there are also some important differences between B cell and T cell development. most obvious of which is that where it happens is different. So where they mature and go through the screening, B cells are going to be in the bone marrow. T cells start in the bone marrow, but then it can happen in the thymus. B cells have to go through the bone marrow, and then they actually will continue their maturation in the spleen. So totally different areas that these are happening between the two subsets of cells. T cells do negative selection just like B cells do, but T cells also have that positive selection they have to go through because they have to make sure that they're capable of binding to MHCs. So they have to have a positive selection and also a negative selection at the same time. The specific arrangements, identities of what gene segments are recombined is dramatically different between the two. And again, we didn't go over that chapter, but those are one area just to keep in mind is different in that process. So where the gene segments are being rearranged. And then what happens when their receptors are stimulated by binding to the antigen is also different. So T cells, remember, they have to require presentation. They can only bind to their antigen when it's presented on an MHC class 1 or class 2, depending on the type of T cell. And they will then differentiate into either helper or killer subsets, depending on this process. B cells, on the other hand, they don't have to have it presented, but they can bind to their antigen, and it can be membrane bound or soluble. They can bind to antigen in a variety of ways, but they do require T cell help to get activated at least the first time, and then secrete antibodies in response to that binding.